Welcome, everyone. Loving all the emojis. This is really a great platform, a fun platform. Could somebody let me know when we're ready to begin?
I think we are ready to start.
Okay, thank you, Erica. All right, hello, and welcome to the continuation of BioConnector 2021. My name is Stephanie Hicks, and I'll be chairing this short talk session on epigenomics and gene regulation within the epigenomics and gene regulation track. We are going to start the talk shortly, but first a few housekeeping items. If you have questions for the speakers, please enter them in the Q&A. You also have the ability to upvote questions to the speakers as they are asked. If you would like to ask your question live, please use the raised hand feature and the moderator will bring you up on stage. There are a variety of emojis, emoticons available to give feedback. If you were in Daniela's keynote talk a minute ago, it was a lot of fun. And if for some reason you need to leave early, a video of the session will be available a couple of hours after the session has completed. I am now going to introduce our first speaker.
Everyone.
Okay. I'm delighted to introduce Marie Jeanmougin from Oslo University Hospital in Norway. And she's going to be talking about positive droplet calling and normalization of droplet digital PCR methylation data. Welcome.
My name is Marie. I'm a project group leader at Oslo University Hospital in the LIMS lab of epigenetics. I would like to thank the organizer for the opportunity to present PoDCall and our package for positive droplet calling and normalization of droplet digital PCR methylation data. So in our lab, we use droplet digital PCR for DNA methylation profiling. Briefly, the technology will partition DNA methylation samples or DNA samples into about 20,000 droplets, where the target and background are randomly distributed across the droplets. An independent PCR reaction is then carried out in every single droplet, allowing the detection of rare events. At endpoint reaction, the droplet containing the fluorescent PCR target molecule are labeled as positive here in blue, while the others are labeled as negative. And the positive droplets are then counted first and then used to quantify the concentration of the target of interest. We obtain such profile where each dot here is a droplet, and on the y-axis you can see their amplitude values. So at the bottom we will have a cloud with the negative droplets, and here at the top the positive droplets with higher amplitude values. And the data can be visualized either as scatter plot here on top or as a histogram of amplitude values distributions. And this technology has emerged as very promising in cancer research. It has been used, for instance, by several to study mutations, copy number variations, or gene expression. But its use to quantify DNA methylation level has been held back by the lack of standardized approach. And especially when it comes to normalization and downstream analysis of the data. So here I will introduce PoDCall, and I would like to highlight two specific features of PoDCall, which are the positive droplet calling and data normalization. So you can picture ddPCR data as a mixture of positive droplet containing the target of interest and negative droplets which show no amplification. As I said, you can visualize this data as scatter plots with on the y-axis all the amplitude values, and here all the different samples that are analyzed at the same time. You can also visualize the same data for each sample, as well as specific histograms. And we have used mixture models to discriminate between the cloud of negative droplets and the cloud of positive droplets with higher amplitude values. And our workflow for positive droplet calling consists in 2 steps and take as input the CSV amplitude files from the ddPCR machine and extract all the amplitude values for all the samples. When running ddPCR experiments, we usually work with 2 channels. One channel with that contains the target of interest, and the other that will contain a reference control for normalization. The first step of our workflow consists in reference calling, where we set a reference distribution, which is usually the methylation positive control, and we test for unimodality of the distribution of this reference. If the null hypothesis is not rejected, we will estimate the density of the single component, test for potential outliers and discard them, and set the reference threshold as the maximum amplitude value. If the null hypothesis of unimodality is rejected, we will estimate the number of components using a likelihood ratio test and bootstrapping, and estimate the density of these components. We will then set the reference threshold after the first component, which contain the negative droplets. What we have observed is that in some cases, there is a shift in the negative droplet distribution across wells. So the second step consists in adjusting this reference threshold for each well to take into account this potential shift. So we will first estimate the shift from the reference distribution for each well, compute the well-adjusted thresholds, and then compute the normalized concentration. As output, we will offer to save the plots, so both the scatter plots and the histograms, and we provide also a result table that I will show you later. So this is an example of how our solution compares to QuantaSoft, the existing solution that is provided by RAD, the main provider of the ddPCR platform. So as you can see, QuantaSoft in some cases doesn't manage to set a threshold, for instance here, between the negative and positive cloud. It is also sometimes sensitive to the presence of negative outliers, which are destroyed droplets. And when we observe these outliers, it will classify all the droplets as positive when we know that these 2 samples, for instance, are negative controls. With PoDCall, we observe that we can manage to set a threshold for all the well. We also take into account and adjust for the negative, the shift in the negative droplet distribution here. And we are quite robust to the presence of outliers, of negative outliers here, and manage to classify these droplets as negative. We have tested PoDCall in different settings, on cell line, on tissue samples, in urine samples as well, with high DNA amount, low DNA amount, large separation between the negative cloud and positive clouds, or more narrow separation, and it performs equally well in all settings. Once the droplet calling is performed, we move to the normalization. We have developed our own alternative to the single locus controls, which are usually used for normalizing ddPCR data. It consists in a panel of 4 genes and is called the 4-plex, and is analyzed in the same reaction as the target gene. We have evaluated our 4-plex against the single locus control at B and C-less and tested them in 34 colorectal cancer cell lines. So here on the right, I show the normalized concentration of the gene CD01 using either the 4-plex, ACTB, or CLES. And the cell lines are grouped here against their chromosomal aberration status. So whether they have losses, whether they have gains, whether they have no loss or no gain, or in the case of the 4-plex, since we combine 4 genes, we can observe both losses and gains. What we observe is that chromosomal aberration here will significantly affect ACTB normalized concentration as well as CLS, while in contrast, the 4-plex remains quite stable to the presence of chromosomal aberration. So the package offers both the droplet calling and the normalization and can be downloaded from Bioconductor. You can run the full PoDCall workflow in R using the wrapper function called PoDCall ddPCR, but you also have access to all the individual functions for importing the data, for plotting, for setting the threshold. And you can also use the package as a user interface using the PoDCall Shiny function. And this is an overview of how the interface looks like. Here you can import the data, import the sample sheet containing all the information on the samples, set the different parameters, and click on the Set Threshold button. You will have access to all the threshold for the target and control, the number of positive droplet, and of course, the concentration for the target, for the control, and the normalized concentration. You can visualize the data either for each well individually as a histogram or scatter plot, or you can visualize the data for several samples at the same time for the sake of comparing them. So as take-home message, I like our robust internal control. The 4-plex has been published already in 2018 in Clinical Epigenetics, so feel free to have a look at the paper for more information. And a patent application has been filed as well for this control. The package is already available and can be downloaded on Bioconductor. An application note will be soon submitted in Bioinformatics, and the patent application has been filed for this solution as well. With this, I would like to thank the whole Lint lab of epigenetics and especially Professor Guro Lint, our group leader. I would like to thank Hans-Peter, who has been instrumental in helping to develop the package, and Heidi, who has been very involved in the testing. I would like to thank our collaborators and our funding agencies, Helse Sør-Øst, the Research Council of Norway, and Kreftforeningen. So please feel free to ask questions. Thanks for your attention.
Thank you so much. So if you didn't see in the chat, we're going to save all the questions for the end, but if you could leave the name of the speaker that you are wanting to ask a question to, that would be helpful. All right, so let's move on to the next presenter. I'm excited to have welcome to the stage Benjamin Tremblay from the Center of Research in Agricultural Genomics in Spain. And he's going to be talking about large-scale analyses of biological sequence motifs with the Universal Motif Package. Welcome.
The Center for Research in Agricultural Genomics in Spain. Before I begin, I would just like to thank the organizers for giving me this opportunity to speak. I'm very grateful. Also, I would like to thank the maintainers of the Bioconductor project. I really think it's an amazing resource, and I'm glad to be a small part of it. For my presentation, I would just like to introduce to you the Universal Motif Package. First, some background. Biological sequence motifs are small repeating patterns in sequences that have some sort of biological significance. For example, they could be binding sites for transcription factors in DNA. In this example, in the top left, I've got some sites aligned, and one way to represent these is as a consensus element. Whenever you have multiple letters at a single position, you can use ambiguity codes to represent those. However, as you can imagine, this can cause you to lose a lot of information when the ratios of each letter are not perfect. That's where the the position probability matrix comes in as a much better format, in which case at each position you record the probability of getting each letter. Now, this is great as a storage medium, it's great for comparison, it's great for visualization, but when you want to scan other instances of this motif through other sequences, a better format to use or type is the position weight matrix. In this case, for each probability, you divide it by the probability of that letter in the background, and then take the log base 2. This ends up with positive scores when the probability is higher than the background, and negative scores when the probability is lower than getting that letter in the background. If you have 0 probabilities, you end up with negative infinity scores, and if that's undesirable, such as if you want to allow for mismatches, then you can add a small pseudo count during the creation of the position probability matrix. One more type that I'll just briefly mention is the information content matrix, which is really good for visualization purposes, and essentially it's the position probability matrix scaled by the level of conservation of each position. In this example, you can also see how easy it is to create and visualize motifs using the universal motif package. This create motif function is very flexible. It takes consensus element inputs, raw sequence inputs, numeric matrices, and you can also create random motifs from nothing. Also, there are other functions for converting from other bioconductor classes and reading common motif text formats. The way that scanning works is that you take your position weight matrix and use this as a scoring system. In your sequence of interest, you break it up into subsequences that are the same length as your motif, and for each letter, each position, you retrieve that respective score in the position weight matrix and sum that up at the end. To get positive hits or hits of interest, you can use a threshold, which can be a certain percent of the max possible score in the position weight matrix, or you can get the probability of getting different scores in your sequences and use the threshold that way. The way to do this in a universal motif package is very extremely simple. In this example, I'm reading in the Eridopsis genome sequence as well as the Eridopsis transcription factor binding sites in the MotifDB package. And then it's just one call to scan sequences using both of those objects. There are many options for controlling the thresholding, as well as the number of threads if you have larger workloads. And you can scan any number of motifs with any number of sequences, and you get very detailed results from that. I'll go over a couple of examples of how you might visualize this information. In this example, I'm reading in some data objects from the package, a set of promoters, a motif. I'm creating another motif as well, scanning those in the promoters. And then using the ggBio package, you can very nicely visualize the locations of those hits. All you need to do is set layout equals karyogram in the autoplot function. You can see how this is done in the ggBio vignette for the karyogram type plots. And you can very nicely see where your motif hits are. A second example is using enriched heat map. In this case, I'm again retrieving the Eridopsis genome sequence. I'm creating a motif known as an ROI motif, scanning that through the genome, and importing some ChIP-seq data as well as ChIP-seq peak location data. Then it's just a call to normalize to matrix, as you can find in the enriched heat map vignettes and enriched heat map itself. And you get very nice visualization of the peak of the ChIP-seq signal on the peaks, as well as the presence of the motifs around those peaks. And in this case, you can very nicely see the correlation between binding of this transcription factor and this motif. Just to expand on this example a little bit, in this case I want to determine whether the enrichment of this motif at these peaks is significant. So I'll just use the enrich motifs function, as well as I'll use peaks retrieved from a shuffled Arabidopsis genome. And using Fisher's exact test, the probability of the enrichment of motifs in the peaks of interest occurring by chance is nearly 0. And just to visualize this again, you can very nicely see how clear the enrichment is in the actual peaks compared to the shuffled genome. One more aspect of my package that I'd like to mention is motif comparison, which is a very important aspect of de novo motif discovery, especially when you want to go back and see if any null motifs actually match it. The way motif comparison works is that you align your two motifs, and position by position you retrieve the letter probabilities and run them through any sort of similarity or dissimilarity metric you'd like. In this example, I'm using the Pearson correlation coefficient. And once you've done that for each position, you sum those up. And if you want to normalize for the number of positions in your comparison, you can just take the mean. Now, comparison can be a bit more complicated, usually, because not all motifs are the same width. So you might need to slide the smaller motif within the larger one. And you might want to allow for overhangs in the alignment. As well, you might want to consider the reverse complement of a motif, which doubles the number of alignments. This can be quite computationally expensive, especially for more complex metrics such as the Pearson correlation coefficient. The way to do this in the universal motif package is, again, very simple. You read in your motifs whatever way you wish, and then once you have them as a list, you call compare motifs. And compare motifs is a very flexible function, has lots of different tweaking settings that are available, different metric methods that are possible. You can go see the function documentation. It's extensively documented. And I've worked my best to make this as performant as possible. So this 800 by 800 comparison matrix takes a couple of seconds on my laptop. And if you have hundreds of thousands of comparisons, as long as you're willing to wait a few minutes, it's not that bad. I'll just mention quickly this merge similar function, which combines steps of comparison and merging, which is very helpful if you have a database that has a lot of redundant motifs. Now, to visualize and explore this comparison output and specifically see if my RY motif might be part of a specific cluster of interest, I'll use the complex heat map package. So I'll just feed the matrix straight to the heat map function, and they get this sort of very nice heat map output. In terms of my RY motif, you can see in the top left there, it might actually be part of a little interesting cluster that could be worth exploring. To summarize, I'll mention the major functions of the package, and would just like to specifically mention the import-export functionality, which I've worked on to make as broad as possible. So even if none of the other functions interest you, I would encourage you to make use of the wide range of import-export functionality to allow other analysis tools to work together that might previously have been incompatible. That was actually the original intention of the package, to serve as sort of universal format between tools. So if you're interested, please go check out the package. I've got several vignettes over on the Bioconductor website. I've submitted these slides to the F1000 BioC21 channel if you'd like to review them. And if you have any questions, please ask or reach out by email later. Thanks.
Thank you, Benjamin. That was great. Okay, just as a reminder, please ask the questions in the Q&A tab. We'll save the questions until the end, and please mention the name of the speaker. So next, I am pleased to introduce Mirko Rugerman from Goethe University in Germany. And he's going to be talking about an R package for defining binding sites called iClip. Welcome.
Get started, and I'm very happy that in the next couple of minutes I can introduce to you a novel R package that we have developed here at the Zanac group, which is specifically designed to help with the analysis of iClip-derived sequencing data. It's also already submitted to Bioconductor, and you can find it in the current developmental version. So the iClip protocol is used essentially to understand protein-RNA interactions on a transcriptome-wide scale. It all starts with the introduction of a covalent bond between the protein and the RNA via UV light in the living cells. And this is then followed up by immunoprecipitation, where we pull out the protein of interest with an antibody, and alongside comes the RNA. And then the RNA is further purified and cleaned and washed until the protein is finally digested, and then the leftover RNA is subjected to reverse transcription. And the reverse transcriptase usually truncates right at this little peptide left over here from the initial protein, which then causes a truncated cDNA molecule, and this is what is then forwarded to high-throughput sequencing. And if you now take the sequenced reads, you map it to the reference genome, you always know that the position right in front of the read start was where the original crosslink in the cells had happened. And by this you get a single nucleotide resolution transcriptome-wide of your protein of interest binding to the RNA. And how does it look like? In principle, what you get as a result is such a single nucleotide resolution crosslink coverage here, where down here are all the positions, and then on top you see these bars, and the higher the bar, the more crosslink events were detected on that specific position. And now people typically use that as an input for a p-calling tool, such as we use here PureClip. And the result of that tool is basically always positions here that are enriched in their crosslinks compared to the local background, which is basically also what you want. But now all of these tools basically deliver this kind of output, and this is where people are left with. And then you can look at the bound targets basically, and that's about it. And we felt that that's maybe not the best way to end it, and there could be more. So what we decided to do is develop here our binding site finder package that iteratively merges and resizes these positions here into regions of unified width that we now call binding sites, while controlling for these called crosslink sites as well as the underlying crosslink event coverage. And how does the package now looks like? In principle, it takes as a core component, it takes all of the relevant data and builds its own data set. So it takes the output of the p-calling as crosslink sites. They are stored internally as G-ranges, and they usually come in as bad files, so you can also nicely work along with these. The crosslink events themselves are stored as RLE lists. Typically, they come in as bigwigs or bad graphs. And then you also need to provide some meta information, such as sample IDs or conditions, for example. And then the central function of this package is basically using the make binding sites function here. That's our workhorse that now uses this iteratively merging and resizing scheme to compute binding sites of the desired width that you can put in. And in order to evaluate if that was what you've plugged in there is any good, you can use the range coverage plot function here that then shows the crosslink coverage over fixed width for the given binding site. And that's what we did here for binding site with 3, 7, and 29. And now you can directly see that here, for example, binding site with 29 is way too large defined, because you include regions here and to the other side as well that do not show an enrichment in crosslink. It rather looks like background. On the other side of the spectrum, binding site with 3 seems to be missing out on a lot of the coverage here, left and right of the gray frame. So that's too narrow. And in fact, 7 here appears to be a reasonably good fit, hitting the sweet spot right where we want it to be. Another thing that we've implemented is check for replicate reproducibility. Because what I didn't say yet is that the initial coverage that I showed was basically the merge of 4 replicates in this case. We merged these in order to boost the p-calling step to find more of the initial sites. But afterwards, we of course need to control for the support of the individual replicates. And that's what we do here. Basically, we count for every binding site how many crosslink events do now overlap a specific binding site from the given replicate. And that results in such a distribution here for all of the 4 replicates. And then you can select a distribution-based cutoff here individually for each of the 4 replicates. So here we decided on the 20% quantile, which then results in a different threshold to be used for each of the replicates given the individual replicate strengths. And lastly, when you now have all these cutoffs, you can, or you basically now can decide how many of your replicates need to meet their specific threshold on a given binding site for you to call this binding site to be reproducible by the replicates. And for this, you can again use the reproducibility filter function here. That function can output a directly finally filtered object that you can then work with, or it can output a matrix that you can directly put into the upset function from the really amazing complex heat map package, which then results in such an upset plot, so you can visualize the results. And in fact, in this data set, it appears to be that most binding sites seems actually to be supported by all of these 4 replicates. And this is basically where the workflow of the package takes you. And from that, you can extract the final individual binding sites and do a bunch of other analyses. And I'll just give some few examples here. One very common is the analysis of the binding spectrum, which, as I said, people typically go ahead and look on the bound targets and then check out the gene types of these targets. And here in that example, it's mostly protein coding genes that are hit by the clip, which is not that informative. But now with the individual binding sites, you can actually drill that down further and look at the bound transcript regions here. For example, looking at how many binding sites are in introns, coding sequences, and UTRs. And in fact, here we observe that most binding sites seem to be in introns, which is, of course, a high information gain compared to just knowing that it's in protein coding genes. And in this example, this also makes a lot of sense if I now tell you that the clip that we're looking at here is from a splicing or intronic splicing regulatory protein. And some further analysis that benefit from individually defined binding sites is like, for example, motive description with any third-party tool, so de novo motive finding with maybe DREAM or STREAM, for example. Also some k-mer counting, k-mer enrichment approaches around these binding sites really work nice. What also can be done is the integration of further orthogonal data, such as RNA splicing, regulated exons from RNA-seq, for example, or additional clip data sets can be incorporated, for example, analyzing how the binding changes in different conditions. And with that, I want to finish and thank all of you, thank the ZANA group. So this package is a joint effort by the ZANA group. There is a recent publication from our group in collaboration with the König group at the IMB in Mainz, where we described the initial processing of the iClip data. And as I also said in the beginning, you can find the package already at the bioconductor developmental version. Yeah, thanks for your attention.
Wonderful. Thank you for that great presentation. Just as a reminder, please add your questions to the Q&A and leave the name of the speaker in your with your question so we know who to ask. All right, and last but not least, I'd like to welcome Catherine Rosen-Gagnon from Rockefeller University in the US, and she's going to talk about a generic R package for clip analysis called ClipFlex-R. Welcome, Catherine.
Thank you. Just 1 second. Okay, so first, I'd just like to thank the organizers for inviting me to present this package, ClipFlex-R. So this is an R software package my colleagues and I developed that provides a complete and flexible workflow for clip analysis. And so as Mirko just highlighted, cross-linking immunoprecipitation, or CLIP, followed by deep sequencing, is a powerful tool to map RNA binding protein interactions with their target RNAs. And so, I mean, he's already gone over this a bit, which was very lucky for me, but to reorient you again to the experimental protocol, in brief, cells are cross-linked with UV light to covalently bind RNA to RBPs, and then lysates are prepared. Then RBP-associated RNAs are purified by immune precipitation. Adapters are ligated for downstream sequencing library construction, and RNA is size selected and purified. And then cDNA libraries are generated and deep sequenced. And finally, in the last step, sequences are analyzed to identify peaks on RNA targets of RBPs. And although the bioinformatic analysis I'm showing occupies only a small proportion of the slide, this informatic analysis of CLIP data is quite time-consuming and laborious. And so I hope you can appreciate from this slide that CLIP is a lot of experimental and informatic work. So why would people do this? Well, it's basically because CLIP can identify transcriptome-wide RNA targets of RNA-binding proteins, RBPs, and this method is therefore a powerful tool to study RNA regulation. And RNA regulation refers to virtually all aspects of the life of an RNA, from its transcription to its transport, splicing, and stability. And there's an intense interest in this field because misregulation has been strongly linked to development and disease. And one famous example is that of spinal muscular atrophy, where both mutations and missplicing of survival of motor neuron gene transcripts leads to deficiencies in protein levels and causes SMA. And because CLIP can be adapted to look at nearly any aspect of RNA regulation, there are many variations on how to generate and how to analyze CLIP data. And these many CLIP variations have been applied to high-profile RBPs involved in disease. So CLIP has really advanced our knowledge of RBP interactions and RNA regulation more generally. And I just want to mention briefly that at Rockefeller, where I'm located, there's a local expertise from the Tuchel and Darnell labs who have focused extensively on CLIP. And so when I began my postdoc, my goal is to apply CLIP to mosquito argonaut or AGO proteins to map RNA interference pathways in a major vector of human disease, the mosquito. And so you've probably all heard of these RNA interference pathways like the microRNA and siRNA pathways. And in these pathways, AGO proteins are targeted to transcript targets by small RNAs. And generally the function of this is to decrease gene expression. And so if you'd like more details on any of this, I really encourage you to check out my recent paper. And so in adapting AGO-CLIP to mosquitoes, I faced several obstacles. So first, as I mentioned, the informatic analysis is fairly laborious, and this is even the case for model organisms. But my use of a non-model organism lacking a pre-built genome blocked me from using many existing analysis tools. And so what are the current analysis tools available for CLIP? Well, biopsy packages include RCAS, wave cluster, and near binding, and each of these focus on a specific aspect of CLIP analysis. And as we just heard in the nice talk previously, Binding Site Finder was also recently developed for peak calling and also shows how to annotate gene and transcript targets using biopsy packages and nicely integrates with biopsy. There are also many GitHub packages. These are mainly focused on peak calling, but there are relatively few complete workflows, and to my knowledge, none are fully executed within R. And so coming into the project, one major pipeline of interest for me was the CLIP toolkit, or CTK. And I had initially started using CTK because it's fairly complete with decent documentation and test data sets. And CTK was also considered somewhat of a gold standard for the specific experimental variation of CLIP that I was performing. And there was also local expertise, of course, using this pipeline at Rockefeller. But in spite of these advantages, CTK is pretty cumbersome, and in some places it relies on pre-installed genomes, which of course is not going to include mosquito. And so now I want to walk you through some of the issues that I had with CTK and how we tried to address them by developing ClipFlexor. So first, CTK relies on many external programs that the user manually installs and configures, which can be quite time-consuming. And so to ease this burden, ClipFlexor now uses the biopsy package Herper to build a Conda environment and automatically install and configure CTK end dependencies within this environment. And so ClipFlexor dependencies would be installed in exactly the same way. And so if you want to learn more about Herper, I highly encourage you to check out Matt Paul's talk on Friday in the infrastructure and programming session. And so once the software is installed, the first step in CLIP analysis is to process our reads. So CTK and most CLIP analysis tools use FastX for at least some processing steps. And what we did was wrap up FastX and CTK processing functions so they can be executed via ClipFlexor in this R software framework. However, it's also worth mentioning that FastX is a very old software and it's not maintained anymore. And so one idea that we had is that by moving this entire CLIP analysis workflow to R, we can start to provide updated processing alternatives like the biopsy package rFastP, and then we can actually test these processing workflows side by side in the same software framework. And the next step would be to map and visualize our reads. So in CTK, this is done using command line BWM Perl. And again, ClipFlexR allows easy access to a variety of other mapping packages in BioC in addition to BWA. And I'm showing you all the packages here. And users can also use our SAM tools and our track layer to manipulate BAM files, make G ranges, and make visualization tracks of their mapped reads. And then the next major analysis step is to analyze target RNAs of RBPs. And to do this, peaks are called, filtered, and annotated, essentially. And here, CTK was a huge issue for me with its preset genomes. So while those working in model organisms can still use CTK functions via ClipFlexR, people using custom genomes can use home or peak calling, which we wrapped in R, or any BioC peak caller of their choice, like the ClipSeq binding site finder package we heard about, or the ChIP-seq MaxR and Seesaw packages. And more flexible annotation is also available by making custom annotation objects and annotating with packages like ChIP-seq or RCAS. Finally, CTK also provides mutational analysis, which one can execute using ClipFlexR wrappers as well. And so finally, CTK and many pipelines lack integrated analysis of small RNAs, and this was essential to my project. So these would be more specialized functions for AGO-CLIP or CLIP of other RNA binding proteins that mediate RNA-RNA interactions. And so when we developed ClipFlexR, we wanted to add flexible quantification of small RNAs and also functions to link small RNAs to transcript targets. So in all, I hope I've shown you that we've addressed some of the major issues of CTK with the ClipFlexR package, and that ClipFlexR does confer many benefits. So it's scalable and parallelized via the BioC Parallel and Futures packages. It's self-contained and reproducible via packages like R-Markdown, REMV, and Herper. And it also reduces user burden and error by easing installation and by using a single software framework to easily integrate outputs from different analysis steps. And so now this package, ClipFlexR, is available on GitHub, and we have documentation and workflows in place here at this site. I'm showing you our workflow that takes you basically from getting your FASTQ off the sequencer to building count matrices and summarized experiment objects that can then be easily fed into other BioC packages. And for example, in this workflow, I show how to annotate peaks in non-model organisms using genomic features and ChIP-seq. And of course, you can perform any other desirable analysis, such as PCAs. And we provide several additional vignettes, which I've outlined here, and we're still working on providing more workflows for users. So in conclusion, I hope I've illustrated to you how ClipFlexR can ease CLIP analysis by easing installation of CTK and dependencies, and also increasing reproducibility. And we've tried to provide R-based alternatives to many CTK functions, as well as R-wrapped CTK functions for comparison of analysis workflows. And importantly for my project on the right here, this ClipFlexR R-based framework and integration with BioC allowed me to call and annotate CLIP peaks in non-model organisms, so in Mosquito, and complete annotation and peak calling. And we also provided new workflows for linking small RNAs to targets. And so we very much hope that ClipFlexR can help other researchers facing similar issues when they're analyzing their CLIP data. And then finally, moving forward, in terms of the structure of the package, we'll likely move to a specialized ClipSeq object as opposed to using summarized experiment objects, as Mirko showed in the binding site finder package. We'd also like to start shifting from outdated tools to updated alternatives, for example, moving from FastX to RFastP processing, and these changes could be implemented and integrated with other CLIP analysis R packages relying on FastX. And so finally, although most ClipFlexR functions are parallelized, there's a few we're still working on, and we'd also like to add more graphical outputs to the workflows we provide. And finally, we of course like to submit to Bioconductor, which we think will be a great way to get user suggestions and feedback, which we very much welcome. So with that, I'd like to thank my colleagues and collaborators who made this work possible, including my mentor, Charlie, and the members of the Rice Lab, as well as the Bioinformatics Resource Center at Rockefeller. I've highlighted here my co-developers of ClipFlexR who are essential to this work, and please check out Matt's talk on Friday. And I'd like to thank my funding sources. So if you're interested in seeing implementation for different analyses or AGO-CLIP more generally, please check out my paper here and the accompanying code. And ClipFlexR is now released on GitHub, and you can find the documentation here. So thank you for your attention.
Thank you, Catherine.
Thanks.
Let's just take a moment and give a round of applause to all the presenters in this session. Thank you so much. I'll add my show of emotion. Okay, so there are a few questions in the Q&A. I'll start with, I think the one with the most votes, from Danya. Benjamin, what is the difference advantage with your motif scanning function versus something like match PWM from the biostrings package?
Well, thanks for the question. So it's been a little while since I've used it, so I might be a little fuzzy on the details, but there's a few differences. One is that the thresholding, the way you decide the threshold in match PWM, you can only do it based on like a fraction of the max score of the PWM, whereas in my function, you can set an absolute score or you can use p-values to determine a threshold. And also, although match PWM is pretty fast, it's not parallelized, whereas mine is, so that's a bit an advantage if you have a lot of motifs. And it very nicely gathers results with extensive information about where the hits are and the contents of the hits into one single data frame or G-ranges object. So it's not fundamentally, it's not doing anything different, but it's more comprehensive function.
Thank you. Okay, Mirko, is the decision for the correct binding site width solely dependent on visual inspection via the coverage plot, or does some kind of scoring scheme exist that tells the user the best binding site width?
Yeah, let's say that's a tough question because it's really hard to define, let's say, what's the true or best binding site width for an RBP, because that usually changes given the cell type, given the RNA that the RBP binds to. But what we do, or what we find is the best way to approach this, is using the coverage on the binding sites. And that can be either done by visualizing that, or we also provide a function that computes you a score, basically by taking the number of crosslinks within the binding sites and then looking left and right, so flanking of the binding site in a window of the same width, counting again how many crosslinks fall in these windows, and then dividing this by the number of crosslinks within the binding site. And that gives you sort of a ratio that tells you how strong the enrichment of crosslinks is within the binding site compared to what is immediately locally in the background. And that helps you in order to guide you through which could be a possible width. And then for these, you can then do the plots that I've shown in the presentation, where you then plot the coverage and then have a deeper look how things actually look visually.
Thank you. I hope there's time for one more question. I think, Erica, can you confirm that we can ask another question? Okay, I'm going to go for it. Yes?
Yes, absolutely.
Okay. Catherine, sometimes during miRNA sequencing, the adapter sequences are still attached to the miRNAs. QC tools like FastQC and the cut adapter used to remove these adapter sequences, does the ClipFlexR also have these QC methods inbuilt?
Yeah, so that's a great question. So if you obtain small RNA or microRNA sequences via your CLIP protocol, yes, ClipFlexR does wrap up processing, pre-processing tools. So we basically follow the CTK pipeline to do that. So it's a combination of wrapped up FastX and CTK. But yeah, in the future, we do hope to provide more alternatives for these pre-processing steps. But there is a way to do that in place currently, yes.
Thank you. Question for Benjamin. Could you suggest a good resource to learn about motifs and the bioinformatics related to it? And thank you for your promising package.
Well, unfortunately, this is just in my experience, in my opinion. There's no one great beginner resource for this. There's a lot of disparate resources for, here's this. And it's complicated by the fact that people have different naming conventions. Like some people say PWM, some people say PSSM, some people say PPM, some people say PFM. So unfortunately, it's a bit of a mixed bag, and I can't really suggest one comprehensive resource. I mean, I do have a vignette that's basically my first slide expanded with the formulas and some basic information, but yeah, that's unfortunately the best I can do. Now, some good resources for motifs in general are things like the memes, the meme suit website, the RSAT website, JASPAR. And if you go there and sort of branch out to publications that are associated with it, then maybe you can sort of trace back history and learn more about it. But sorry, I don't have a great answer.
Okay, I'm going to quickly look at the chat. One question I have for all the panelists, I'm just curious, I think some of you touched on it. In terms of the objects that you're working with in your packages, what's the availability or the accessibility to be able to work with, for example, downstream data visualization packages such as ggplot? I saw that in Universal Motif, you mentioned the use of ggbio. Are there easy ways to leverage some of the downstream visualization tools that might be helpful for you, or?
Well, in terms of visualizing the sequence hits, then that was, basically the best I can do is, or the best that the package does right now is you can get a granges object, and then there's a host of packages on Bioconductor that use granges objects as the input for visualization, and I showed a couple examples. For motifs themselves, so I do output the visualization of motifs as a ggplot object. So you're free to use any of the, cowplot, for example, or patchwork to create more complex visualizations with the motifs. There's one other aspect. Oh, and I also have a lot, I didn't go into it, but there's also a lot of sequence utilities like calculating the background of sequences. And in one of my vignettes, I do have an example showing how you might compare sequence composition using that information, which is just a nicely formatted data frame. And so that very easily plugs into the ggplot or the tidyverse workflow. So there's nothing maybe specifically geared towards that, but I've made it so that the outputs are as clean as possible so that you can pick how you would visualize them.
Thanks. Any other responses from the speakers?
I mean, I would say very similar to Benjamin. Like, I didn't design anything specifically in mind, but I did try and make outputs that are easily integrated with different visualizations. But I think that's like a great place for us to further develop the package, actually, and something we're hoping to expand to. But you can, of course, get outputs that are easily fed into existing visualization packages. Yes.
Great.
And in principle, we are doing also the same, like having the output in a way that it can be plugged easily into ggplot2, for example, on the one side, and then on the other side, not visualizing, but rather continue to work with it. It comes in the form of the granges then, which is then again widely open to what is used in Bioconductor. And maybe another thing is that at some point we try to put out matrices in a way that they can easily be picked up by complex heat maps, so that you then get also easy visualization of that.
And it's the same for the PoDCall package. We actually have our own tools for the visualization, but we also provide both in R, but also on the Shiny application, way to upload the results as data frames, and it's easily used for any kind of visualization downstream or any kind of analysis. So we have tried to make it as clean as possible as well.
Thank you for all your responses. I don't have any other questions with that, unless there are other questions. Lots of praise in the chat if all the panelists haven't, or all the speakers haven't seen it. I will say thank you to all the attendees of the session. And thank you very much to the presenters. Great talks.
Thank you very much, everybody. I just want to remind everybody the networking session is starting now, so you can head on over to the lounge and join a table and chat with some of your fellow attendees. And the speaking program will resume in 90 minutes.